about mold-lactic fermentations. As you know, in nature, fermentation is brought about by a collective action of large number of microorganisms. These may include fungi as well as bacteria. Fermented food and beverage can also be produced as a result of extensive microbial growth. Fungi and bacteria mediated fermentation of food item takes place under natural condition, and these are also exploited at commercial levels. Fungi or molds secrete hydrolytic enzymes that degrade the complex biopolymers. Lactic acid bacteria constitute a ubiquitous and heterogeneous group of bacteria capable of fermenting a variety of carbohydrates with the production of lactic acid as one of the major end product. Molds, which are commonly known as the fungi and the yeast, are widely used for fermentation of food either singly or in conjunction with lactic acid bacteria. Molds liberate glucose and other fermentable sugars from complex carbohydrates. This process is termed as saccharification. For instance, mold lipases and proteases convert the complex fats and proteins into simpler form. They also contribute to the synthesis of flavor imparting compounds. Mold enzymes such as amylases, proteases, lipases, glucooxidases, pectinases and tannases are also produced in the food. Species of mold, Rhizopus, are capable of producing fumaric acid, lactic acid and other organic acids, and they also produce ethanol as they excrete out zymases. Examples of mold mediated fermentation are manufacture of soya milk, cheese in Japan by using Aspergillus oryzae fungus. In Finland, the production of villi by fermentation is mediated by a mold, Geotrichum candidum. In mold lactic acid fermentation, there are two components. One is the lactic acid bacteria and other are the molds. Let us know what are lactic acid bacteria. Lactic acid bacteria constitute a ubiquitous and heterogeneous group of bacteria capable of fermenting carbohydrate with the production of lactic acid as one of the major end product. The homofermentative lactic acid bacteria are acid producers. For example, Lactococcus lactis, subspecies lactis, Lactococcus lactis, subspecies cremoris, or acid and flavor producer like Lactococcus lactis, subspecies lactis biovariety diacetyl lactis, Streptococcus salivarius, subspecies Morphilus. The heterofermentative organisms are capable of producing lactic acid, carbon dioxide and aromatic compounds like ethanol and acetic acid from glucose. The members of the genus Bifidobacterium ferment milk sugars to acetic acid and lactic acid, yielding a distinctive vinegar taste. Among the microbial constituent in the mold lactic acid fermentation, there are lactis. Lactics include rods, for example, lactobacillus and cocci such as Lactococcus, Streptococcus, Leuconostoc and Pediococcus. The non-lactics are bacteria such as Bifidobacterium, Brevibacterium, Acetobacterium, Propionibacterium. The fungal components, mainly the yeast, are Candida and Cluveromyces, Torulaspora and Saccharomyces. And there are also eukaryotic molds like Penicillium, Aspergillus oryzae, Mucus species and Geotrichum candidum. Let us see some of the examples of mold-lactic fermentation in dairy industry. Some of the cheese such as washed rind cheese are a product of mold-lactic fermentation. In this, the cheese are periodically cured in a solution of brine or mold bearing agent, which include Brevibacterium linens, Debaryomyces hansenii or Geotrichum candidum. These organisms impart a strong flavor as the cheese matures. The second group is smear ripened cheese. In this case, older cheese are smeared on the young cheese to transfer the microorganisms. Example include Munster and Port Salut. There is another group of cheese which are known by their color, and they are blue cheese. These cheese are formed as a result of action of a fungus, Penicillium roqueforti, which is the key microorganism responsible for the blue color of the blue cheese. As the mold grows, the blue veins develop in the cheese, giving the product its characteristic flavor. Examples are Stilton, Roquefort and Gorgonzola cheese. There is another group of cheese which are known as soft ripened cheese. It requires mold for the ripening process. The inoculum consists of Penicillium camemberti and lactic acid producing organism Streptococci. Example of this type of cheese are Camembert and Brie. A number of cheese are produced by collective fermentative activity of mold and lactic acid bacteria. Some examples of mold-lactic fermented cheese are blue cheese, smear ripened cheese, soft ripened cheese and washed rind cheese. Let us learn a little detail of the blue cheese. Blue cheese is produced by fermentation mediated by the mold Penicillium roqueforti. This mold produces lipases that converts the fat in the cheese to free fatty acids and methyl ketone 2-heptanone. Methyl ketone 2-heptanone is the chemical which is responsible for the characteristic blue aroma. The blue pigment seen in the blue cheese is produced by the action of fungus Penicillium roqueforti during the sporulation process. Another example of mold-lactic acid fermentation is acidophilus milk. This product is also produced as a result of the collective activity of the lactic acid bacteria and fungus. In this process, the milk is heated at 90 to 95 degrees C for a period of 10 to 15 minutes. This is followed by cooling it down to 35 degrees C, and at this stage 3 to 5% starter culture comprising of Lactobacillus acidophilus and the yeast Saccharomyces lactis are added. The first stage of fermentation takes place at 35 degrees C temperature, and at this stage the acidity of the milk is 0.8% lactic acid. The milk is bottled at this stage. The second stage fermentation takes place at lower temperature of the range 10 to 17 degrees C for a period of 6 to 12 hours. The final product is stored at low temperature and it is subsequently consumed. Another popular mold-lactic fermented milk product is VILLI. VILLI is a mesophilic fermented milk found in Finland. The starter used for preparation of VILLI comprises of lactic streptococci, Lactococcus lactis, subspecies lactis, Lactococcus lactis, subspecies cremoris, Leuconostoc mesenteroid, subspecies cremoris, biovariety diacetyl lactis. The mold employed for the fermentation are Geotrichum candidum. The interesting feature is that Geotrichum candidum mold is symbiotic with lactic streptococci. In the production of VILLI, the molds are producing carbon dioxide in the milk to form carbonic acid and carbonates. The lipolytic activity of the mold while growing on the surface increases the concentration of monoglycerides and free fatty acids, which impart typical flavor to the product. Mold-lactic fermentation are also used for production of probiotic beverage like KEFIR. KEFIR is an acidic, alcoholic, fermented milk product. The starter cultures used for the preparation of KEFIR is a symbiotic mixture of lactic acid bacteria, yeast and acetic acid bacteria that are present together in a polysaccharide matrix. The symbiotic fungal yeast partners belong to Zygosaccharomyces species, Candida species and Saccharomyces species. The starter culture is used as KEFIR grains. These KEFIR grains include members of Lactobacilli and Streptococci, Lactobacillus species, Leuconostoc species, Lactococcus species. KEFIR grains have predominance of lactic acid bacteria, acetic acid bacteria, yeast and a fungi Geotrichum candidum. After learning about dairy mold-lactic fermentations, let us now see some of the prominent examples of non-dairy mold-lactic fermentation products. The first product is TEMF, where soybeans are fermented by the fungus Rhizopus oligosporus and lactic acid bacteria. The other product is miso. Miso is produced by the action of yeast, mold and lactic acid bacteria on soybean. The fungus commonly employed are Aspergillus oryzae, Aspergillus species, lactic acid bacteria, Pediococcus species, Lactobacillus species and Lactobacillus plantarum. Sourdough is another example of mold-lactic fermentation. Here, lactic acid bacteria and yeast ferment the cereals, millets, pseudo-cereals, cassava flour to yield sourdough. Jiku is also produced by the collective action of mold, yeast and bacteria, and they ferment the starch-rich food. Mold commonly employed are Aspergillus oryzae and Rhizopus oryzae. Amylolytic yeast Saccharomycopsis fibuligera and Lactobacilli collectively mediate the fermentation process. The non-dairy mold-lactic fermentation are also employed for fermentation of meat, resulting in the product which are commonly known as sausages. The organisms used in this fermentation are homofermentative lactobacilli, Pediococcus which produce acidity in the sausage. The fungal partners are Penicillium chrysogenum and Penicillium nalgeovens, which ripen the surfaces, which are also responsible for developing the aroma and also improve the texture and reduces the ripening period. These fungi, Penicillium and mucus species, contribute to the sensory qualities of the product. Therefore, sausages are produced by the collective fermentative activity of lactic acid bacteria. Therefore, sausages are produced by the collective fermentative activity of lactic acid bacteria and molds. Students, let us summarize the fermented foods which are produced by the combined activity of lactic acid bacteria and molds are called as mold-lactic fermentations. In this fermentation process, both fungi and lactic acid bacteria, by virtue of their metabolic activities, produce compounds that complement each other so that the resultant products are with desirable qualities. Mold-lactic fermentations are utilized widely in many dairy and non-dairy foods, and they are commercially exploited to produce various food products. Some examples of mold-lactic fermentations include cheese, sausages, tempeh, miso, soy sauce, jiku, sourdough, acidophilus or yeast milk, kefir, villi, komes, etc. These products are rich in nutrients. They also possess extended shelf life and they are functional foods. They are promoting the health of the consumers. Thank you.